which part of this schematic do we already recognize? Okay, we did glycolysis, so we did glucose to pyruvate. What else? We did talk about oxaloacetate, but we didn't quite do this part yet. We definitely did this one, and we did the Tca cycle with that lactate.
We did the transamination.
We read about this part, so we did the fact that alanine can actually be a byproduct of pyruvate breaking down to alanine, and then the nitrogen going off to the urea cycle. We didn't do the urea cycle yet, and we didn't do the amino acid portion of that yet, but that will be to come. So here we are at pyruvate. So now we're going to need to go through and do some of this, make it acetyl-CoA, so we'll do that reaction today, and then we'll use acetyl-CoA to go into the Tca cycle, and we'll see now a new cycle this time, but a cycle that doesn't have a beginning or an end. It's just, now it's going to be a loop. So when we start with this, we can't do the urea cycle or the Tca cycle yet. We have to do one reaction before we get to the Tca cycle. So we need to convert pyruvate to acetyl-CoA before we can do this. So this is actually a picture, a little micron picture, a really small picture of our complex called pyruvate dehydrogenase complex. And so pyruvate dehydrogenase complex is 8 times 10 to the 6th kilodaltons. So that's what? 8 million kilodaltons, which is 8 billion daltons. So that's huge. I mean, that is like absolutely enormous. But the reason why it's so big is actually because it's made up of 3 different enzymes all together. So typically when you're looking at these metabolisms and you see the word complex, it usually means there's more than one enzyme that's all together in some sort of 3-dimensional picture, and that's going to do the reaction. So there's a little bit of inconsistencies in what people call this. So they usually will call the pyruvate dehydrogenase complex, and then within that there is an enzyme called pyruvate dehydrogenase. So the complex means not only the pyruvate dehydrogenase plus the other 2 is what it means. And then when you look at each individual one, then we'll see that there are 3 different enzymes in there. So the purpose of this is we're going to take our pyruvate and we're going to convert it to acetyl-CoA. We need a little A on the end of that. And carbon dioxide. So remember, pyruvate has 3 carbons. So one of our carbons comes off as carbon dioxide. And the other 2 stay as part of the acetate part of acetyl-CoA. So pyruvate's got 3 carbons. One of the carbons comes off as carbon dioxide, and the other 2 carbons are with the acetyl-CoA part. And this is going to be a reaction that happens in the mitochondria. So it is an extremely complicated reaction. It requires 3 different coenzymes: TPP, lipoamide, and FAD. And the delta G for that reaction is a negative 8 kcals per mole. So you knew somewhere along the way we were going to need a FAD or an NADH. How did we know? What up there gave us a clue?
So mitochondria?
More.
Is there lots of NADH in the mitochondria?
There is. So the clue is this word. So these dehydrogenase reactions, when you remove hydrogen from them, typically they're going to go on to FAD or NAD+, and that's where those hydrogens are going. And this also gives you an indication that it's a redox reaction. And so when we think about FAD and NADH, we should think about redox reactions. So I said this was made up of 3 different enzymes. So the first enzyme is the pyruvate dehydrogenase, the individual pyruvate dehydrogenase, not the complex. So we're going to label that one E1 because it gets very complicated in our picture when we try to do the little mechanism of it. And its coenzyme is TPP, which is thymine pyrophosphate. And the second enzyme in that big, huge complex is called dihydrolipoyl transacetylase. And the last one is the dehydrogenase. So E1, E2, and E3. And we've got TPP, lipoamide, and FAD are the 3 different coenzymes, one for each of the 3 enzymes in our big pyruvate dehydrogenase complex. So we are going to need a few more cofactors. What else do we need? That's not in that little list.
Didn't the TPP and pyruvate dehydrogenase take like the magnesium?
Yes, but that's not going to be one of our coenzymes.
Like the acetyl-CoA.
Yes, exactly. So we need some coenzyme A. And even though our NAD is not going to be showing up in this part of it, it is going to be present in, and actually that should be NAD+. And it's going to be one of the coenzymes that's going to be part of the products that we get. So it actually is not part of the coenzymes for the 3 enzymatic reactions, but it is going to be required for a component in solution in order for us to generate that acetyl-CoA. So a lot of people, I have about 2 different kinds of schematics for the process. So this one, let's look at this one really carefully. There's another one that's a little bit easier to see. I like this one because it has the 3 different enzymes there in a somewhat stepwise mechanism for going from the pyruvate to the acetyl-CoA. So where should we start when we look at this schematic?
On the pyruvate on the left.
Right, let's start with pyruvate. All right, so here's pyruvate. So when we look at the overall reaction, it just goes pyruvate becomes acetyl-CoA and kick off a carbon dioxide. Not so fast. So the first thing that does happen is that carbon dioxide comes off with our pyruvate dehydrogenase enzyme. So, but what is actually the purpose of this? Tell me what that little structure means to you.
Didn't the PPP make it more soluble because it has all the oxygen on it?
This is actually not going to float away in solution. This is going to stay bound to the enzyme. So think about this part as the active site. So where you have a coenzyme sitting in that active site. So pretend like, oh my goodness, pretend this whole circle was our enzyme. And we've got this active site sitting right there. And so when I label these, and I labeled this one C1, where is C1 in this pyruvate? Right, this is C1 right here. So we've cleaved this bond. But what have we done to those other two carbons? Yes, we did a little rearranging. So if we label this one C2 and C3, where is C2 and C3 up there in our active site?
Two's in the middle.
Right, 2's in the middle. So C2 has the alcohol, and C3 is still a methyl group. So what is that enzyme doing? What is the enzyme with the coenzyme doing?
Taking the place of the carboxyl group.
Right, it's taking the place of the carboxyl group. So basically what it's doing right now is just holding on to C2. So you had 3 carbons in solution. You did a decarboxylation reaction, released 1, but in the process you left these 2 carbons attached to the first enzyme. And that's why the rest of these enzymes have these little swirls attached to them. Because what is it actually doing now? So we did this, TPP by itself, went over, combined with these 2 carbons, the C2 and the C3, and are now attached to pyruvate dehydrogenase. Then what does it do? What is this little swirly one doing right here? What are these green ones doing?
The 2PP comes up and binds, that carbon binds to the sulfur on that group.
Right, so TPP exchanged away for our nice little, our lipoamide, our lipoamide. So this little coenzyme now gets the 2 carbons. And now what happens here? What did we do to get this one? I had to break a bond. What bond did I break? I could find one. So I definitely broke this bond. And what bond over there did I break?
This.
Right, between the 2 sulfurs, right, so I broke that bond.
It's not, is that a disulfide?
Yes, it's a disulfide. So this lipoamide cofactor has a little disulfide in there. And when that disulfide popped open, instead of it being an SH, one side is an SH group, the other side is now making a sulfur C-double-bond-O bond. So this TPP passed off the 2 carbon units. So when it's in the pyruvate dehydrogenase, it's an alcohol. Then when it gets passed over to the lipoamide, it's actually the ketone. So the advantage to this part so far right now, that's our acetate group. We're pretty close ready to pass it on off to coenzyme A. So we passed our 2-carbon unit onto our lipoamide. So now we make this sulfur acetate group just like it is in coenzyme A, except this one's attached to lipoamide at this point. So now, when we go and join our coenzyme A, at this point, the coenzyme A that's going to come in has a thiol group right there in the reduced form. So it's got the SH on it at this point. So when the coenzyme A joins with the lipoamide with the little acetate group on it, pass off the acetate group to coenzyme A. And so what would I label this carbon now? Is this carbon 2 or carbon 3?
Still 2.
It's still 2, right. This is C2, and that's C3. This is C2 and C3, right, in fact. Okay, we should be done. So why are we not done? So why are we not done? Because I'm almost here. I started with pyruvate. There's one of my products. And there's the other one of my products. So what's the whole purpose of that stuff? Why do I need that third enzyme, the dihydrolipoyl dehydrogenase enzyme?
That way, with some energy.
Close.
The NADH, which is good.
Right, so one of the things is we want to produce the NADH, but we can't produce it directly. So we kind of produce it via an intermediate. So after I've given up the 2 carbons, now my lipoamide at this point is in the reduced form. So I've got 2 hydrogens attached to that lipoamide. Now, in order for me to regenerate that disulfide, what do I have to do to the hydrogens? Right, I need to take those hydrogens off. Now, if I'm smart, I can couple that to generating a really awesome FADH2. So, but the cofactor for this enzyme is only FAD. So I can't take this reaction and shove it over here because it's not the correct coenzyme for it. But if I just do another little handoff of those hydrogens, I can generate an NADH and a proton. So if I labeled these 2 hydrogens, this hydrogen here and that hydrogen there, trace where those hydrogens go now.
Go on the FAD.
Right, so they're going to go on the FAD to become FADH2. And then the very last step, which is a non-enzymatic step, that NAD plus will take off those 2 hydrogens. One of them will go onto there. Oops, sorry, there's 2 of them in there. 2 of those. Like that.
So those 2 hydrogens are the ones on the NADH.
Correct.
Okay.
So the overall reaction looks really simple. So we start with pyruvate and an NAD plus. Actually, there's technically 2 NAD pluses. generate a carbon dioxide. We need carbon dioxide, we need acetyl-CoA, and 2 NADHs. Oh, that looks so simple. Not so much. And 1 pyruvate dehydrogenase complex. But this reaction happens all on the exact same little, it's almost like a square. It's a little cube-shaped enzyme complex. So it's all happening in the exact same space so that this doesn't get a chance to diffuse away. It stays right in the complex and carries right on through all the way to the NADH stage.
With NAD plus that whole portion at the top, would that happen kind of simultaneously as this part, so that it would kind of like meet almost?
Exactly. So this, once you get to the edge of this one, now you're exposed to the cytosol or the solution, the solution, and now you can be exposed to that NAD+. So the only thing in there that's actually diffusing is the NAD+ and the carbon dioxide. And the coenzyme A comes in as a diffusion also.
So how did you say that active site doesn't diffuse away?
This one? So this TPP is actually bound to the enzyme.
Okay.
Or at least a portion of the enzyme is holding it in with hydrogen bonding.
Okay.
So if the enzyme is holding the TPP and the TPP is covalently bonded to those 2 carbons, it won't diffuse away. And that's a common theme as we get towards doing lipid synthesis. When you get these big complexes, you have to kind of tether them and hold them so they don't diffuse away.
Is that really the only reason the TPP is needed?
Yes.
Okay.
And the TPP is needed because it needs to hold this so that the lipoamide can come in and make that new bond right there. This bond wouldn't be able to glom right onto that one. We can't go straight from there. We need to go to the alcohol first, and then we go from the alcohol to the ketone. So this is the one from our textbook. So it has some advantages to it. It's okay. You can kind of wind your way through all the eras. But there are a couple different versions of it, and this other version I found seems to be pretty good also. Has some advantages. So that's the one on the next slide. So this one has some advantages and some disadvantages. The advantages, it's prettier, it has some nice colors in it, but it's also kind of just doing this blob that each of the enzymes is a blob. All right, so when we look at this one, where do we start? Okay, let's start up at the top with our pyruvate. And so when we go into this one, we can see our TPP. There's our TPP. And you can see the TPP is bound to enzyme 1. So that's our pyruvate dehydrogenase. So this one bound, so now we have TPP and our alcohol. And you can see that enzyme 2 is ready. It's got the lipoamide disulfide ready to do the reaction. And so when you're, and this one is good because it does see that FAD is actually bound to E3, just waiting. Not doing anything yet, but waiting. And then when we go to make our sulfur carbon bond, we have our reduced thiol. And then when our coenzyme A comes in, we release our 2-carbon unit. And then we generate the dithiol is the best way to describe that. And then since our FAD is right there, it's going to become FADH2. It oxidizes that back to the disulfide. And then we can give our hydrogens to NADH, and then we can regenerate our TPP, ready to do the reaction again. So this one's, no, there's some advantages to that one. So when we look at this one, there are multiple steps. So if we do a description for what's happening at step 1, we would say the pyruvate loses its carboxyl group. It's going to bind to the TPP and wait for the TPP enzyme complex, wait for the lipoamide. Then when we're over here at step 2, now we've got our lipoamide, and we're going to be ready to make a new sulfur carbon bond. So the hydrogen atom and the acetyl group are transferred to the oxidized form of the lipoyl group. So we're going to break that disulfide on coenzyme 2, on E2, I should say. And then we're going to react with our coenzyme A to produce our acetyl-CoA, and it leaves 2 thiol groups ready to be acted upon by our FAD, pick up those 2 hydrogens, and then transfer the 2 hydrogens to NAD+ to make NADH. I think all 5 of these are in there. Okay, so all the descriptions are in there, so you can just read them. So this is what we have for our overall reaction. So we have an NAD+ and a pyruvate, and a coenzyme A, and we've gotten a carbon dioxide, an acetyl-CoA, an NADH, and a proton. And actually, we can do that same one up here too. I'm going to look back at our book and see why they wanted to put 2 of them in there. Because this reaction only generates 1. So I'll go back and look at our book as to why they did 2 of them.
Is this still plus the complex too?
On the arrow.
Yeah, the arrow, yeah, that's over the entire complex, yes. So that arrow gets pyruvate dehydrogenase complex over it. Okay, so obviously we have to do a regulation. So if we look at the top of the next slide, we're going to look to see what happens right here. So this is our pyruvate dehydrogenase, and add the word complex to the end of that because it is all of the reactions. So this is pyruvate dehydrogenase complex. We've got all of our parts moving here and there. So what should we say for the influence of acetyl-CoA on that pyruvate dehydrogenase complex? What do you think, plus or minus?
Minus.
Minus?
Because we're making it, so if there's nothing, then it would shut it off.
Exactly. So that's definitely going to be a minus. So what do you think about NADH?
I think it's also minus.
Minus again. 2 minuses on those. So the acetyl-CoA part is a feedback inhibition, and NADH is in general it's a feedback because it's not actually part of that. It is the end of result, but the FAD is actually what's part of the complex, but it's in general. It's very similar to a feedback. So these are going to both be inhibitors. Now, acetyl-CoA is actually a competitive inhibitor. So So we have our two regulation components. So which one do you think is going to be most affected by these inhibitors? So I think I put them on. And so what do you think? Where do you think coenzyme A is going to be the inhibiting? Okay, so E2. So why do you think E2?
Because that's where it would make it
True. Any other possibility? What about structural similarities? So what else kind of has that structure to it?
For the lipoamides.
Mm-hmm. What else does? The only thing is it's missing a carbon.
Yeah.
Put a carbon with coenzyme.
You pyruvate. So this group, this portion, actually binds to the same portion that those two carbons from pyruvate bind to. So it's actually inhibiting E1. So the pyruvate dehydrogenase complex, or not the complex, the pyruvate dehydrogenase enzyme E1 is actually where that's going to bind. So it's doing feedback inhibition on the first enzyme, which kind of makes sense. Because if you don't want the rest of it to go, stop the first one. So remember, when it's competitive, it means that it has to bind to the same active site as pyruvate does. So there must be some sort of structural, spatial relationship between these two components, these two carbons in the coenzyme A, and those same two carbons in pyruvate. So what do you think about NADH?
That would be the third one, right?
Yep, that's a great one.
Can I ask a quick question about the other one though? So it's competing with the TPP then basically?
No, it's competing with pyruvate.
Oh, okay.
So remember, TPP is a coenzyme.
Okay.
So, but pyruvate is the substrate. So the acetyl portion of the coenzyme A is competing for the binding spot. So remember when we did enzymes and we did competitive and non-competitive. So if our substrate is the triangle shape, a competitive one would be something that looks really, really similar to it. And it's going to bind in the same spot, not exactly, but it's in a spot such that it's going to not allow the pyruvate to bind. So it's competing for that same active site. So which has to be structural similarities between the acetyl portion of the inhibitor and the pyruvate portion of the substrate. Does that make sense? And so another form of regulation is phosphorylation, dephosphorylation, our favorite. We see it all the time. So if you look at the next slide, we're going to go and try to figure out, let's see, we did those two. And here's our enzyme. It's called phosphoprotein phosphatase. But anyway, be careful. Make sure that your notes have this arrow fixed. It is one of the few typos in the book. So this arrow needs to be going up, pointing up towards the inactive form. So we need to be putting pluses and minuses down for all of these components. So first let's talk about what's happening right here. So what's the difference between the pyruvate dehydrogenase at the top and the one on the bottom? Right, so as soon as you phosphorylate it, now you're inactive. So the phosphorylation is on the serine of E1. Which means, where is the serine? So remember the side chain for serines, alcohol, right? So if we put that phosphate group on it, phosphates are big compared to that CH2 group. So where must this serine be? Where on E1?
By the active site.
Yes, by the active site. Somewhere in here. Because when you put that big, huge phosphate group in there, it's so big, it doesn't allow pyruvate to bond. So that serine must be in the active site for that pyruvate dehydrogenase enzyme. So basically, the phosphate group is like a little plug. So leave it there until you want to generate a bunch of acetyl-CoA. So pyruvate dehydrogenase, phosphorylated, just sitting there, hanging out, and ready to be inactivated. Or it's staying inactivated, ready to be activated. So then when you remove the phosphate group, inorganic phosphate goes off into solution, and now you have the active form, and on it goes to produce NADH. CO2 and acetyl-CoA. All right, so now let's decide. What about the enzyme that turns it from the active form to the inactive form? So we are using a phosphate from ATP and generating ADP. So this phosphate group that's on here came from ATP. And this kinase enzyme is the one that's going to make it inactive. So let's decide. Let's do the logic of those. So NADH plus or minus on this kinase?
Did I activate it?
Okay, so you say minus.
What I'm asking. So this is it an activator or inhibitor to inactivate the enzyme?
This for this kinase.
Okay.
I got a lot of activating, inactivating. So I'm going to, so if I have high levels of NADH, let's just do the logic. If I have a NADH, do I want more NADH? No. So how do I stop it?
Phosphorylate.
By phosphorylating it and inactivating it. So what do I want here?
Phosphate.
So when I do acetyl-CoA, another plus, right? So what do you think about coenzyme A that doesn't have any acetate attached to it? Minus. Because it's for the exact same reason that since this one is plus, when I don't have the 2 carbons on it, the answer would be minus. So if I want to stop this inactivation, this means I have plenty of coenzyme A around. Go ahead and make some acetyl-CoA. You're okay to go ahead and make it. All right, so what about NAD+? So in our reaction, NAD+ is on the left-hand side.
So minus.
Correct, minus. Think about ADP.
If it's like dephosphorylated ATP, so you lost ADP.
So we should think about this. If this gets too high, right, we don't want this to continue, so we need that one to be minus. And what about pyruvate? Minus what?
So you could combine
So I knew exactly what you were thinking, though. So it is going to be minus. So because this is the substrate for this pyruvate dehydrogenase complex and this first enzyme, then if we have a high concentration of it, we should probably slow it down because we don't want to have too much of it.
Can you explain ADP again?
So ADP is inhibitor and on this particular enzyme because when you've done this conversion to ADP and the concentration of ADP starts to rise, it means that this needs to slow down because this conversion right here, you're going too fast. So what other indicator could it be? So you could say it is the it would be like the opposite of too much ATP. Okay, are we good on those? So we had to do this whole reaction just to get to do the TCA cycle. So we prepped our pyruvate that came from either glycolysis or the breakdown of amino acids. So we have this pyruvate now. Now we've made acetyl-CoA, and now our acetyl-CoA as a 2-carbon unit can go into the TCA cycle. And so we generate some energy right there. We generate an NADH so that if we start to tally up how much energy are we getting out of glucose, we have to include that NADH as that little in-between reaction between glycolysis and TCA. So some people call it the Krebs cycle, same thing. Citric acid cycle, TCA cycle, all of them mean the exact same thing. I think they should just pick one and stick with it. So when we did glycolysis, we recovered 5% of our total 2840 kilojoules per mole. So we're like, ugh, that was pretty bad. Or small. So, but if we continue to go through the glycolysis out to pyruvate, through pyruvate dehydrogenase complex, and through the TCA cycle, we're going to be able to generate a lot more. So by the time we get done with the TCA cycle, we'll tally it up again. So our citric acid cycle, we're going to get 2 more ATPs out of the citric acid cycle. 2 direct ATPs out of the cycle. And the rest is going to come from oxidative phosphorylation. So when we did the electron transport chain, we said that NADH joins in pretty early in. So how many ATPs can we get from an NADH through the electron transport chain? Remember? It was a blue slide and had a bunch of arrows on it. And then we had delta G values going down the right-hand side. And we said, oh, that delta G is so big we can make an ATP out of it. So when we did the NADH, there's 3 of them. And when we did FADH2 jumped in after NADH, so you couldn't get that 3rd ATP out, you could only get 2 ATPs out of it. So remember, when you see NADH and FADH2, just think just as good as ATPs. So justifications that you can use for ATP, you can use for NADH and FADH2 as well. So we're going to do, so we have our pyruvate, and it's passing into the mitochondria. And our pyruvate dehydrogenase complex is right here on this arrow. So we can release our carbon dioxide, release our acetyl-CoA, and now we're ready to do the citric acid cycle. And so while we're in the mitochondria, we've got NADH that's going to be produced, our FADH2, NADHs, and we can do the electron transport chain in there as well. So when we do the overview, it's kind of a cool reaction, or We're going to start with 2 carbon units. So we know we're going to start with this acetyl-CoA right here. So these 2 carbon units are going to join into the cycle. And they're going to join into the cycle where eventually we're going to produce a carbon dioxide and NADH. But in the process, we have to do about 8 different reactions just to do that. So this will be our first cyclic reaction. So it's not linear like glycolysis and gluconeogenesis are. It's actually in a circle. Oops, sorry. Anything you have to write anything on. Okay. So, oh my goodness. Let's see what we got. All right. Pyruvate dehydrogenase complex. We just did that one. We've got our acetyl-CoA right there. And so here is where we're going to join in. Our 2 carbon unit is going to join in with our 4 carbon unit. So our oxaloacetate right here is a component that's already ready to join in with our acetyl-CoA to make citrate, and then we'll go through the citric acid cycle. So this citrate synthase is going to be the first enzyme that we see in the Tca cycle. So this citrate synthase obviously is going to be one of the ones that we're going to do really high regulation on. And we've already seen oxaloacetate. Where did we see oxaloacetate already?
In the mitochondria. We just did gluconeogenesis.
Right, and gluconeogenesis was one of those bypasses. It was like bypassing 1. We saw malate also. So we saw malate, and we saw this little malate transporter. So we could convert it to, we did the carboxylate pyruvate to oxaloacetate, gets passed out as malate, and then we can go finish gluconeogenesis out here. So we have a little crossover, a little series of crossover reactions right there. So that little conversion for malate, oxaloacetate, and pyruvate, it's got 3 different pathways that all interconnecting into that little small little 3 organic component of conversions. So when we look at this one, it's a nice way to follow our carbons before we jump into all of the names that are for each of these components. So we've got our acetyl-CoA with our 2 carbons. And so when our oxaloacetate joins in, it's a 4 carbon unit. And our first our 6 carbon unit will be citrate. And so what happens as we go through this, we're going to see our citrate will be converted to another 6 carbon unit. So we're just going to be changing the bonds in it. And then what happens when we do step 3? So where did our first carbon go? Right, our first carbon left is carbon dioxide, and at the same time we can generate an NADH at that same step. Then we have a 5 carbon unit, and it goes down to a 4 carbon unit, and we've gotten rid of the second carbon. And then for the whole left side of the cycle, we stay a 4 carbon unit. Kind of odd. So we stay 4, but we generate a GTP. We stay 4 again, but generate an FADH2. We stay 4 again and generate an NADH. And this is the malate oxaloacetate conversion right there. So this side of the reaction is lots of chemistry just so that we can get enough delta G values to generate an FADH2 or an NADH. So as the result of 1 turn of the cycle, we're going to get 3 NADHs, 1 GTP, 1 FADH2, and 2 molecules of carbon dioxide. But how should we tally this up if we were talking about starting from glucose?
Would you have more ATP?
So remember when we do this pyruvate, we've got 3 carbons. How is that related to the glucose that we started with when we went to glycolysis?
6 carbons.
Right, so when we generate, the last bit of glycolysis generates 2 of these pyruvates. So we generate 2 pyruvate for every 1 glucose molecule. So while this is good, it has to specify that this is only 1 turn of the cycle. And if you try to do, biochemists do this all the time, they love to try to tally stuff off. Like, how many ATPs would you get out? They love it. I don't know why they love it, but they do. But it does help so that if you're doing a glucose, and out for glucose is 2 pyruvates. So 2 of those pyruvates goes through the pyruvate dehydrogenase complex twice. So you generate 2 carbon dioxides, 2 acetyl-CoAs, 2 NADHs, and your 2 protons. Then you've got 2 acetyl-CoAs to do the math of this one. So you could take all of this and double it, and that would be equivalent to doing a glucose for only the TCA component. Because if you're doing glucose, we need to tally up the parts. So if we do... So if we go through glycolysis, and I'm just going to do the carbon parts now. We get 2 pyruvates, and then our 2 pyruvates have to go through pyruvate dehydrogenase complex. And that will generate 2 acetyl-CoAs. Then you have to do your 2 acetyl-CoAs through the Tca cycle twice. And that generates right there. There's 2 of those plus 2 carbon dioxides. Don't lose our carbons here. And then we get 6 carbon dioxides. Weird. It's weird to know that if you tally where those glucoses go, they actually just become carbon dioxide. But in the process, we do all the other metabolism.
Why is it 6 and not 4 for CO2?
So we're going to do 4 from TCA plus those 2 from the pyruvate dehydrogenase complex. Good. All right, we'll stop there for today. Continue on with TCA on Thursday.